Today's lecture will be on the anatomical structures of the skeletal muscle fiber and the importance of these structures as it plays a role in the sliding filament theory. The first place that we'll begin the discussion will be on the sarcolemma. This is the outer shell or the plasma membrane. This cell membrane of the muscle fiber surrounds the sarcoplasma. So the sarcoplasma is going to be contained within inside of the sarcolemma. The next structure is going to be the transverse tubules. I'll point it out here at the bottom. And then also it's located in this top picture as being this green tubular network. This is where we see the action potential, which is going to travel along the sarcolemma, and then it's going to spread internally first into these T tubules. So we would have the sarcolemma covering in this bottom picture, and then the action potential will first enter into the green structure or the T tubule. That action potential will then circumnavigate this individual myofibril. The next structure is going to be the sarcoplasmic reticulum. I'll circle it in the top picture. It's this tubular network that's in purple. And then in the second picture here, this is the sarcoplasmic reticulum. And the importance of the sarcoplasmic reticulum is it forms, again, a tubular network around each individual myofibril, and its most important aspect with the sarcoplasmic reticulum is that it's going to store calcium. The other component that we see associated is going to be these terminal cisternae. These terminal cisternae, which are located on each side of the green structure of the transverse tubule, is again another tubule of sarcoplasmic reticulum enlarged, fused, and forms expanded chambers called terminal cisternae. These expanded chambers on the outside of the T tubules, they are a reservoir of calcium ions, which again are important for initiating muscle contraction. If we see in this description, we would see two terminal cisternae, one located on the right and one located on the left, and then the T tubule, which is in green. If we add these structures together, it forms what we consider to be called a triad. Again, when we're looking at the functional component or the contractile unit of an individual myofibril, we are going to see that it is called a sarcomere. And inside of these sarcomeres, we're going to see the protein filaments, or what we consider to be called the myofilaments of actin and myosin. So the first area that we should begin our discussion is going to be just the structural component of the thick filaments. This is also called a myosin. It has two components. The first one is going to be, or each strand, so we see here this whole structure is going to be a thick filament. And then off of this thick filament strand, we're going to see the component of the myosin molecule. Each strand has a free globular head, so this would be the free globular head, and an attached elongated tail. And both of those components would make up one single myosin molecule. On the globular head, we see this is an important location for ATP binding site. And this ATP binding site is during contraction, these myosin heads are going to reach up towards a thin filament, and they form a cross bridge by binding to the actin of the thin filament. And it will require energy in the form of ATP. When we're looking at the thin filaments, or what is also known as the actin, and the F just means filamentous as far as definition. The actin or thin filaments is composed mainly of two strands of twisted around each other. They are going to be, each one of these is going to be either the tropomyosin or the troponin. So the first one that we should look at is going to be the tropomyosin. So the tropomyosin, located here, this tropomyosin has, the green structure has a thread-like protein that is going to cover portions of the actin. And the portions of the actin, you can see it here, is a binding site that is going to be important with the cross-bridge theory. The troponin, when we're looking at troponin, the troponin located here has a calcium binding site, which is one key importance, and it also attaches to actin and tropomyosin. So it's also binded to the green cord of tropomyosin and also the different forms of actin. So each one of these actin strands, when we're looking at just the actin strands, is going to have the G-actin, which G stands for globular, and then also a filamentous actin, which means that it is a row of G-actin, and then when they're combined as a row, we call this a filamentous or an F-actin. So in this slide, we can see the two components of the thin filaments. We have the G-actin, individual G-actins, or the row of G-actins, which is the filamentous actin or F-actin. We have the troponin, located in blue, and then we also have the tropomyosin. Again, the troponin is important because we'll have calcium binding to troponin, and it's also connected to the tropomyosin, which when calcium binds, we'll see that how it forms or opens up the activation site. With the thick filaments, we have the globular head, which has, of the myosin fibers, which has the ATP, and then we also have the elongated tail that's attached to the thick myofilament. These two important structures, when we see the G-actin, or when we see the globular head reaching up to attach to the activation site of the actin filament, then this will form the cross bridge. And important to note when we're looking at the M-line is just a protein that helps stabilize the position of the thick filaments. During the sliding filament theory, these thick filaments will stay stationary. The thin filaments will move closer together after the binding site or the cross bridge between myosin and actin have been achieved. When we're looking at the different components of a sarcomere, so the first thing is a sarcomere, or the contractile functional unit of a muscle fiber, goes from a Z disc to its Z disc. And these Z discs that run perpendicular to the myofilaments, and they serve as an anchor point to the thin filaments. So you can see here how each one of these thin filaments are connected and anchored into the Z disc on both sides. The next area we'll concentrate is going to be the A band. So the A band is the central region of the sarcomere that contains the entire thick filaments. So we can see here that drawing a line down from the end of one myosin to the other end of the other myosin, this region would be considered to be the A band. The A band does not change length during contraction. Again, these thick filaments are going to remain stationary. We're going to see the thin filaments moving closer to the M line during contraction. The next area we'll focus on is going to be the I band. And the I band contains only thin filaments. So we can see here we have a thin filament, and on the other side we have a thin filament. So from I band, this central region, we're only going to see the thin filaments. So it extends from one sarcomere, from the A band of one sarcomere to the A band of another sarcomere. As mentioned in the previous slide, the M line, this region is important because the M line helps stabilize the position of the thick filaments. And finally, the last area is going to be the H zone. So when we're looking at the H zone, this is on either side of the M line, and it only contains the thick filaments. During maximum contraction, this zone will disappear when the thin filaments are going to be pulled over the top of the thick filaments. So during contraction, the H zone will actually disappear. So as previously mentioned, when we see cross-bridging, the first slide is going to show the bicep brachii in a relaxed state. So we'll have the relaxed sarcomere or Z disc to Z disc. We'll see that the thin filaments have not moved towards the M line. And then during a concentric contraction of the bicep, we're going to see this is going to be considered to be a fully contracted sarcomere. So the first things that we should notice is that disappeared when we look at the bottom picture versus the top picture. The first thing that disappears is the H zone gets a lot smaller. The I band also, when we're looking at it, gets smaller. So we see a decrease in the H zone. We see a decrease in the I band, and this is during contraction. We see that the zones of overlap, so this area here, get larger. So we see the overlap of the thin filaments over the top of the thick filaments across the M line. Those are going to, zones of overlap are going to get larger. The Z lines are going to move closer together, or what we consider to be the Z disc. So we can see in the top picture the distance of the Z disc versus the contractile sarcomere. And obviously the second picture we see a closer Z disc to Z disc orientation. And the only thing that stays relatively the same during contraction is going to be the A band. So when we're looking at the A band, again, the A band is only going to contain the areas of the thick filaments. And since the thick filaments are not moving during the sliding filament theory, we do not see any change to the A band. So this area remains what we consider to be constant.